Let's talk about brain rejuvenation. And there's been also a lot of exploration of partial reprogramming in the brain as well, including by ourselves. One of the earlier studies from an Serrano's group, who's been a pioneer of partial reprogramming, as I showed in 2013, they've continued looking into it, and they showed that partial reprogramming can improve memory on the object recognition test. And this is a very cool result, very recent result, a 2024 paper by Andronech group from Stanford, showing that partial reprogramming can increase or induce neurogenesis in old mice, like basically generation of novel neurons in the hippocampus of old mice, which has been a very kind of controversial aspect. Can we have neurogenesis in adult brains? And at least with partial reprogramming, it seems that we can. Another brain reprogramming reprogramming paper from Rodolfo Goya's group in Argentina, showing that if you deliver a gene therapy into the hippocampus of old rats, in this case, you can increase their cognitive performance. And this is very similar to what we've done at YouthBio. We've delivered a gene therapy into the hippocampus of old mice and also Alzheimer's mice, and we also see positive results. And Rodolfo Goya's group saw improvements in cognitive tests and also saw a reduction in epigenetic age of the rats treated by partial reprogramming. They also done a different study on female fertility, where they injected female rats in the hypothalamus with partial reprogramming therapies, and they showed that after inducing partial reprogramming in that brain region, that has positive effects on female fertility. And moving on to Alzheimer's, my personal passion, and of course the therapeutic focus of YouthBio, from the very beginning we had good reasons to believe that Alzheimer's is a good indication for partial reprogramming, mainly because Alzheimer's has a strong epigenetic component to its etiology, to the way Alzheimer's happens. And basically, gene expression in the brain cells of Alzheimer's patients can explain why the disease progression happens. As you might remember, since epigenetic changes are reversible, we can be, at least with some confidence, expect that if we reverse the pattern of gene expression in the neurons of Alzheimer's patients, we can expect, if maybe even a reversal, definitely slowing down of Alzheimer's symptoms, maybe even a reversal of Alzheimer's symptoms. This was the theory. Now we have evidence in practice that this indeed seems to be the case. This is a study in an Alzheimer's mouse model showing that you can epigenetically prevent Alzheimer's. Then this is actually the study from 2019 that was the main inspiration for the hypothesis I just outlined, basically showing that you can epigenetically reverse Alzheimer's symptoms in a mouse model. And basically show that epigenetic modulation can not only prevent the symptoms, but it can reverse them. And this was what made me very optimistic that in patients, we can observe the same reversal of symptoms using epigenetic modulation by partial reprogramming. And this is the paper I gave a preview in the beginning. I was very happy to see this. This is a 2023 late result, basically validating the same observations that we had at YouthBio, that partial reprogramming in a mouse model of Alzheimer's disease can have beneficial therapeutic effects on Alzheimer's mice, Alzheimer's symptoms. And also not only at the cognitive testing level, but also at the level of biomarkers, they showed that you can have lower levels of beta amyloid, which is a key biomarker of Alzheimer's disease, lower plaque burden in these mice. And also on the cognitive tests, of course, those mice also showed better performance in those tests. And these observations of this group validate our observations, which were very similar. We saw a reduction in beta amyloid, and we saw an improvement in cognitive tests in the mice. And I briefly wanted to go over our results. Our main approach was, our main tenet behind YouthBio is that we need to be cell type specific, that you have to have specificity in terms of triggering partial reprogramming in different cell types. And this was validated by Alejandro Ocampo, who, if you remember, was a pioneer of partial reprogramming. And he's also our collaborator, but he showed that if you avoid the liver and the small intestine, you can push reprogramming for up to 10 days of consecutive expression of reprogramming factors, and all of mice survive. But if you don't, if you keep reprogramming the liver and the small intestine, then the mice start dying after day 4. And basically, this necessitates the cell type specific approach to reprogramming, that you have to avoid certain tissues. And this means that from our standpoint, we can be also targeted in the cells that we induce partial reprogramming in, with the different cell types listed here. And of course, neuronal cell types are our first priority, with Alzheimer's being the first indication. And there's, I already mentioned, there's good reasons from a scientific standpoint, there's good reasons from regulatory standpoint, why Alzheimer's disease is a good indication for Alzheimer's. And our first animal study was a proof of concept to show that we can deliver our therapeutic constructs to the brain and activate them in the brain and get the expression of these factors in the brain. And once we validated that, we went into 3 different disease models, with Alzheimer's, progeria, and age-related cognitive decline being 3 models, and we saw positive results in those tests. Switching kind of our focus from the past to the future, the important factors in translating partial reprogramming, they're listed on this slide. And I think one of the main aspects is delivery. And this is really the last interesting aspect of the presentation that I'll go into, because a lot of people think delivery to the brain is a insurmountable challenge, but actually there's a lot of precedent in Parkinson's, and Jean already actually talked about this, in direct delivery of therapies to the brain, including cell therapies and gene therapies. And this slide lists like a dozen different gene therapies that have been directly delivered to the brains of Alzheimer's patients by direct injection into substantia nigra. And so also, this has been shown in Alzheimer's disease as well. There you can deliver into the hippocampus, or this just very close to the hippocampus, gene therapy. And this was a 2013 study, which then they had a follow-up like 7 years later, showing that the patients that had this injection into their brain had sustainable expression of the delivered construct for like up to 7 years, which makes me very optimistic that our constructs that we deliver to the brain can also be expected to be very long-lasting. And this is another paper showing the same precedent of direct delivery into the brain. As I mentioned, this just sets the precedent that this delivery is possible. But of course, invasive delivery is not great. If we can avoid sticking a needle into patient brain, we should. And for that, there's now this concept of ultrasound-guided delivery, where it's a non-invasive method of getting things to the brain. And this table lists like many different mouse models of diseases in which this approach has been tried. And beyond mouse models, it was also demonstrated in patients already. This was the Toronto Sick Kids Hospital study that showed that you can have successful delivery into the brain of pediatric patients using ultrasound-guided delivery. And I won't get into this slide just for the interest of time, but basically future directions of partial reprogramming, they're listed here. I think we're well on our way of finding novel factors. Some companies are dedicated to finding novel factors that are not as risky as Yamanaka factors. YouthLib and YouthBio, their main mission is to find new factors, and I think we'll need tissue-specific and cell-type specific factors that will be most effective in a given cell type. And for that, you also need cell-type specific delivery mechanism, which I think the field is also exploring. And so in closing, I just want to say that I'm very optimistic that both partial reprogramming and brain rejuvenation by partial reprogramming will get to the clinic very soon. And I just want to close with this quote from Dr. Belmonte after they published the seminal Ocampo paper: "With careful modulation, aging might be reversed." And I believe that with partial reprogramming, we're well on our way to figuring out just how to reverse the aging process. Thank you very much. And